Imagine trying to do something simple, something you don't even think about, like drinking a glass of water, and you just can't.
Right.
You can't do it without spilling. You can't sign your own name. Eating soup in public, forget it.
That's the reality, the daily grinding reality for millions of people with essential tremor. E.T.
And the scale of this problem is, it's just staggering when you look at it.
It really is. We're talking about 7 million Americans right now. That's more people than have Parkinson's disease in the U.S.
Oh, wow.
And here's the kicker. For almost 60 years, since 1967, they have had exactly one approved drug. And it's barely effective for many.
6 decades of neglect. It's unbelievable. And that's what makes the news we're diving into today so, seismic.
Absolutely.
Just yesterday, December 29th, 2025, the FDA granted Breakthrough Therapy Designation to a new drug, Ulixacaltamide.
And the phase 3 data is, from what we're seeing, pretty stunning. Some sources are calling it statistically bulletproof. This isn't a small step.
No, this could be a genuine revolution for these patients.
It could be. And that's our mission today, right? To be your filter, to cut through the biotech hype and get to the reality.
Because there's a huge conflict at the heart of this story.
A huge one. This exact class of drug has failed before, and this specific drug, Ulixacaltamide, its own phase 2 trial, it missed the primary endpoint. It failed.
So that's the multi-billion-dollar question we're asking. Did the company, Praxis, actually learn from these failures and design a better trial? Or did they just keep trying until they got a result they liked?
That's the tension, and it sets our agenda perfectly. We're going to break down Ulixacaltamide across 4 pillars: the biology, the clinical endpoints, which is where all the drama is, the manufacturing, or CMC, and finally, the regulatory path.
All right, let's start with the battlefield itself, essential tremor. It's so much more than just shaky hands.
It is. It's a progressive neurological disorder that, it just chips away at your quality of life and your dignity.
Things we all take for granted.
Completely. You can't put on makeup. You can't shave. You can't sign a document. It robs you of your independence. And it gets worse year after year.
So what is actually happening deep inside the brain to cause this? Give us the simple version of the biology.
Okay. It all centers on a kind of a highway in the brain. It's called the cerebellum-thalamocortical circuit.
The cerebellum, that's the coordination center, right?
Exactly. And deep inside it, you have these very important cells called Purkinje cells. Think of them as the brake pedal for that entire circuit. They regulate and dampen signals.
Okay, so they keep things smooth.
They're supposed to. But in essential tremor, these cells are dying off. The brake is gone. And so the whole circuit, this whole loop, it just starts running red hot. It's hyperexcitable.
Short circuit.
A very specific kind. The neurons stop firing normally and start firing in these weird synchronized bursts. Yeah, a rhythm. An oscillation between 4 and 12 hertz. And that electrical bursting in the brain translates directly into the rhythmic shaking you see in someone's hands.
So if the circuit is running hot, how does this drug, Ulixacaltamide, cool it down? What's it blocking?
It's targeting a very specific piece of equipment in the neuron called a T-type calcium channel, the Cav3 family specifically.
And those channels are the problem.
They're the enabler. They are what allows that burst firing to happen. They let calcium rush into the cell, which triggers this repetitive rebound bursting. It's like the spark plug for the tremor.
And Ulixacaltamide just jams the spark plug.
Precisely. It's a selective blocker for those channels. No calcium influx, no rebound bursting. The theory is really elegant. You stop the bursts, you stop the tremor.
And the early data in animals looked good.
Very good. You can induce tremors in animal models, give them these blockers, and the tremor improves by, 50, even 80%.
So why are we only grading the biology a B+? If the theory is so clean, why not an A?
2 big reasons. First, human genetics. You'd think with so much ET running in families, we'd find a genetic link to these T-type channels.
But we haven't.
We haven't. There's no clear human genetic proof that this is the fundamental cause. It's a bit of a gap.
So it's a plausible theory, but not a proven one.
Right. And second, the history of failures. Another company, Cavion, had a similar drug, CX-8998. It was abandoned. It just didn't quite work. So hitting this target is clearly hard.
That B+ makes perfect sense then. It's a solid bet, but it's not a sure thing.
Right.
And that shadow of past failures leads us right to the clinical trial data. Because the phase 3 results are why we're even having this conversation. They are spectacular. Lay out the numbers from study 1.
Okay, study 1 was big. 473 patients, 8 weeks. The result on the primary endpoint was an improvement of 4.3 points on something called the MADL-11 scale.
And the p-value?
A p-value was less than 0.0001.
Okay, for everyone listening who isn't a biostatistician, what does a p-value that low actually tell us?
It tells us that the probability this result happened by pure chance is less than 1 in 10,000. I mean, in this field, you just don't see numbers like that very often. It means the effect is real and robust.
Statistically bulletproof.
It's as close as you get.
And then there was a second study, right, to look at durability.
Yes, a really elegant design called a randomized withdrawal. You take patients who are responding to the drug, and then you either keep them on it or switch them to a placebo.
To see if the tremor comes back.
Exactly, and it did. 55% of patients on the drug maintained their improvement. For those switched to placebo, only 33% did. It shows the drug is what's causing the effect, and the effect lasts.
Now, what's so critical here, you mentioned it, is the endpoint they used, the MADL-11 scale. Why is that specific scale so important for the FDA?
Because it's a patient-reported outcome, a PRO. The FDA is very skeptical of what we call surrogate endpoints, like a reading from an accelerometer on a watch.
They don't care about the number, they care about the person.
Precisely. They want to know, did the drug actually make the patient's life better? And this scale measures 11 real-world tasks. Can you write your name? Can you use a spoon? Can you drink from a cup? It's all about function.
So let's translate the data. The difference between the drug and placebo was about 3 points on that scale. What does a 3-point improvement mean in human terms?
It's life-changing. It's the difference between I can't hold a coffee cup and I can drink coffee normally. It means you can sign your name again. It's regaining a piece of your independence. That is what the FDA calls clinically meaningful.
That's powerful.
Yeah.
But we have to talk about the shadow. The A- grade for the endpoint is because while phase 3 was great, the phase 2 trial, Essential 1,
It failed.
it failed completely.
Yep.
P-value of 0.126. They used a different scale, and it just didn't work.
And this raises that critical question. Is this a story of smart science where they learned and optimized the trial? Or is it a story of endpoint shopping where you just run trials until one hits?
Because you can do that in pharma, right? If you run enough tests, eventually one will come up positive just by chance. Why should we trust this result over the last one?
It's a fair challenge. The reassurance comes from two things. First, the phase 3 trial was just better designed. It was larger, and they enrolled patients who are probably more severe, which can make the drug's effect clearer.
Okay.
And second, and this is key, all the secondary endpoints were also positive across both studies. If they were just endpoint shopping, you'd expect to see the primary hit, but other measures would look weak or inconsistent. Here, everything pointed in the same direction. They optimized, they didn't cheat.
All right, let's move on to logistics. De-risking the future. You can have a great drug, but if you can't make it, it's useless. Let's talk CMC chemistry, manufacturing, and controls.
This is maybe the easiest part of the story. Ulixacaltamide is what we call a small molecule. It's a simple oral tablet you take once a day. This is category 1 manufacturing. Think aspirin.
So contrast that with something like a complex biologic or a gene therapy.
Oh, it's night and day. Biologics need living cells, giant sterile bioreactors, a cold chain for shipping. It's incredibly complex and expensive. This drug is just made with standard chemistry. It's stable at room temperature. The CMC risk is about as low as it can get. We're giving it an easy A.
And the potential reward for that low risk is astronomical.
I mean, 7 million patients with no real competition. Analysts are talking about peak revenue of up to $14 billion a year. That's a massive market.
That kind of potential with minimal manufacturing risk is, well, it's the biotech dream scenario.
It is, and they were smart. They already used their commercial-scale manufacturing process for the phase 3 trial, so they are locked and ready to go. Supply will not be an issue.
So let's talk about the final hurdle, the regulatory path. That Breakthrough Therapy Designation they got yesterday is a huge deal. Why did the FDA grant it?
Because the bar was incredibly low. The current standard of care, propranolol, is from 1967. Yeah, and it only works for about half of patients. And of the other half, many can't even take it because of side effects or contraindications, asthma, heart block. There is a massive, serious unmet need. This drug fills that gap.
So what does that designation actually get them in practical terms?
It puts them on the fast track. More meetings with the FDA, more guidance, and most importantly, eligibility for priority review.
Which cuts the review time from 10 months down to 6.
Exactly, and they already completed their pre-NDA meeting.
For the listener, what does a pre-NDA meeting really signify?
It's the last big meeting before you submit everything. It means the company and the FDA are already aligned on what the application needs to look like. It's a huge de-risking step. No surprises.
So given all that, what's the timeline we're looking at for approval?
They'll submit the application in early 2026, let's say March. The 6-month priority review clock starts, and that puts a potential approval right in the fall of 2026.
When the FDA does its final risk-benefit calculation, what's that going to look like?
I think it's pretty straightforward for them. On the benefit side, you have 7 million patients with a huge unmet need and a drug that shows clear, life-changing functional improvement.
And on the risk side?
The safety profile looks remarkably clean. 0 drug-related serious adverse events in the trials. Just mild, manageable things like dizziness. The benefit just massively outweighs the known risk here.
So you're confident. Approval odds?
75–80%. It's a solid A for the regulatory pillar.
Okay, let's wrap this up.
Let's do the final synthesis. 4 pillars. Biology. We gave it a B+, a plausible mechanism, but it's shadowed by history.
Endpoints. An A-. Really meaningful results in phase 3 that overcame that phase 2 miss.
Right. CMC, manufacturing, a solid A, low risk, massive commercial potential, and regulatory, another A. Breakthrough designation, FDA alignment, high confidence.
So the overall verdict from this deep dive is...
Approvable, confidence level, I'd say 80%.
This is the first new mechanism for essential tremor to succeed in phase 3 in nearly 60 years. For 7 million people, that is. That is profound hope.
It is. We still have to acknowledge that lingering skepticism, the phase 2 miss, the need for longer-term data. But what we have right now is transformative.
And it raises a final question for you, the listener, to think about. We know the drug dampens the tremor circuit. It's a symptomatic treatment.
Yeah.
But will it be truly life-changing in the real world, or just incrementally better than what's already out there?
And that's a perfect transition, because while Ulixacaltamide is a traditional small molecule solution, the next frontier is something else entirely. Gene editing. We've got a deep dive coming on precision biosciences. They're also targeting a breakthrough designation for their gene editing approach to hepatitis B.
And the question we'll be unpacking there is a big one. Is their RCS editing system really safer than the better-known CRISPR system? Or is that just brilliant marketing spin on some very complex tech? We'll be digging into that next time.